We're live.
Welcome to the Late Sesh, everybody. I got the announcement duties today, so I just ran with it. Hopefully that was on time because, I don't know. I don't have any indication whether we're live or not on my screen.
I think you
What?
did.
But anyways, no, I don't have any. Nothing tells me
Top
whether we're
left?
live.
It doesn't tell you?
Doesn't say live up there for you and give
Hold
you
on,
like
hold on.
down.
I do see it now. Yep. Okay, I see it
Oh
now.
my goodness. I can't believe you've gone this long. Oh, that's why it's like, yeah, that's why I was like, you can do it while I take this dab. It'll show you when we're live and you start. But I didn't know you didn't.
Yeah, I didn't see
Okay.
that. I didn't see where it was. I had it
All right.
minimized down smaller because
Perfect start to a late sesh, guys.
Mm-hmm.
So,
Cheers, everybody.
cheers, chat.
nailed it.
I'm going to reheat this banger.
Cheers, everybody in chat. Cheers, anything grows. J. Patrick, Madam, and Scruffy Old Bugger in chat already that I see.
Cheers, chat.
Everybody's piling in now.
Oh, that was the wrong. I don't know. That was the wrong link, though.
Oh, I thought I was legging out or something when your screen went black. That was actually what happened.
Yep, I definitely pushed the wrong link.
Pushing
One of the
buttons.
push buttons.
Little technical difficulties. How's everybody doing?
We may have a special guest tonight, thanks to Tara.
I think she'll be with us.
I'm excited for that.
She has a kiddo. She's probably needing
Yeah.
something mom duties, I'm sure. But yeah, super excited that Rachel from Deep Roots will be with us tonight to talk about tissue culturing again and answer any questions and give us some knowledge, fill our heads.
I sent her the link and said, come on whenever. So she'll probably just drop in.
Oh, that's
So, Chad, if you want to get your tissue culture questions, mushroom questions, she's a mushroom head. We found out. I had a bunch of sex tests done by her, and that was successful. So, shout out to her business.
That didn't sound weird.
How many
It
plants,
always sounds weird when you talk about sex tests.
how many plants did you have sex tested?
30.
30?
I've never heard anyone say I had a sex test and it was successful. That's why I said that sounded weird.
Well, have you ever heard anybody need a sex test?
Did you pass the sex test?
Some of them did. No, well, I don't know. No, there is no pass or fail.
So you had
It's
30.
just female or male.
How many of those were females?
There are 3 results. Thank you. There's also an not determined or not. I don't know where it is, but I've
There's
gotten that
zero
third
none
result.
determined.
I've gotten that
Zero
before,
none
and it
determined.
ended up being a female.
Oh, nice. I got 6 out of like 10 that I sent in, but the others were for my buddy.
Oh.
And he got a lot of females. But he's one pack I found out he was just double checking.
Oh,
So he was making sure it was all females.
okay.
But yeah. So that's the Indigo Daydream I sent in.
Mm-hmm.
So I got 6 of those that are female. And I got one more IPM spray, and then they're going to flower. That's what I was cleaning my tray today, getting it all ready.
Exciting.
Yeah, I'm finally gonna be able to. I never use my irrigation system. I got the Net
What?
FM.
Mm-hmm.
I'm always hand watering because everything's different. Everything's always like the other tray right now.
Oh,
There's one plant disco fries. That's on day 54. 2 plants shredder and Mac 1. They're on like day 30 something. Then 2 more plants Cap Junkie and Raphael. They're on like 21 maybe. So I have to hand water. It's just so much easier to hand water all that. They're all different
yeah.
sizes too. Like some of them I just like the Raphael. I was just sick of it. It was growing so slow in veg. Clone, getting flower. Like I just want to see. I want at this point. I just want to see if you're worth anything to me.
Yeah.
So that one's tiny, so I can't just throw the Netafim sprayers in and just hope for the best. I'll overwater it even in cocoa. But so that's why. But yeah, so this will be all one strain, all at one time. I'll be able to plug my Netafims in, just turn the sprayers on for at least half my flower room there, or down here in the basement. But yeah. So I got all those cleaned up. Oh, there she is. Welcome, Rachel from Deep Roots. Glad you can
Hi.
make it.
What's up, Rachel?
Hey,
Not
Rachel.
much. How are you guys doing?
Good.
Good. Glad you could make it.
Yeah, thanks for having me back. I appreciate it.
Appreciate you.
Yeah, we were just talking about the indigos I got tested by you.
Oh, nice. How's
Yeah.
your hunt going?
Well, they're still in veg, but I think number 4 is smelling pretty good. So
It's a
we'll
good sign.
see. Yeah, I cloned them all. Got like 2 or 3 clones. So I'm putting them in flower this week. But we'll see.
Nice.
Yep.
Good luck.
I'm excited. Yeah. So any exciting news on tissue culturing? I saw you post a HHVLP plant or whatever it is.
Yeah, hop implant.
That came through your place?
Yeah, I mean, I work with a lot of people that have hop latent thyroid in their facilities or in their gardens, all the time. Yeah.
Oh.
Most
You
of
do,
the time, though, when people get positive test results, they kind of like chuck the plant because they don't want it to risk any of the other
Yeah.
plants getting contaminated. But this one I was able to get and do some experiments on before the person threw it away. Which, I mean, theoretically, I get so much material and I could infect a plant, probably anytime.
Yeah. That's interesting. So yeah, there wouldn't be anything you could do to get it well, you could clean it?
Yeah, meristem culture should clean up hop latent thyroid. I mean, you really have to make sure you're getting a clean dissection, and your methods are good so that you're not reinfecting your culture yourself. Just like with your garden, you don't want to reinfect your garden with contamination from the outside world. But yeah,
Okay.
meristem culture
Yeah.
should be eradicating any viruses, anything. It should be getting down to the bare essentials of the plant. You're getting just basically like a stem cell portion of the plant. So it's just
Okay.
a ball of cells that can develop into anything at that point.
Oh, all right. Well, anything, or well, then what do you mean, like a new pheno?
No, I mean like stems or roots.
Oh, stems or roots. Okay.
Like any
All
kind
right.
of plant tissue. I should have specified. Sorry.
Yeah. I don't know anything about the microscopic stuff. But that's interesting. That was a good explanation. A ball that can form into anything.
Yeah, it's just undifferentiated, just like stem cells.
Oh, yeah. I was reading, or saw a thing that if you eat dark chocolate, you can double your stem cell
Yeah.
count. So
Oh, interesting.
it
Really?
was like a little bit a day or something. Obviously you don't want to eat too much, but
Yeah.
real dark chocolate though, you have to get good real dark chocolate.
So
Yeah.
it must be the cacao or whatever that's in chocolate.
Yeah,
What is it?
something
What
like that.
specifically in chocolate has anything to do with the stem cells? Did they say? What was the
No, it
association
was
there?
typical 15-second video, but
Chocolate has a ton of
Oh, you read a headline.
polyphenols.
You're telling me.
Chocolate
What
has
was
a
that,
ton
Rachel?
of
I'm not sure.
polyphenols.
Oh, polyphenols. Okay, so also is coffee and things like that.
Yeah, exactly. Which is why coffee also has good health benefits as long as you're, having clean coffee and you're using, water that's relatively clean. When they process coffee, there's a fermentation process and it can grow mold. So I'm sure certain coffees have more mold toxins than others, and they can affect you depending on how your body reacts. But, like for the most part, it's a healthy thing, as long as you're not drinking it all day and all night.
yeah. So I googled it, Spartan. 2 cups of real cacao hot chocolate a day for 30 days showed an increase in stem cells. So
Oh, we
this
gotta
is
get that
pro hot
chocolate, man. We gotta
chocolate.
try that
That's why
out.
I remember it, because I went, ooh, hot chocolate. I'm a big fan of hot chocolate. So maybe I got mad stem cells. Never know. But I also like probably the fake chocolate. So it says not
Yeah,
the
the
milky,
sugar is
sugary
especially you
variety.
like.
And that's the stuff I love. But I do have some boxes of dark chocolate. That's interesting. So
There's a good question in chat from Anything Grows. If a plant contracts something, can it be more recurrent down the line? Or once it's gone, it's gone? Like, would it be more susceptible to that disease again,
Oh,
even after you've
it should
cleaned
be
it?
once it's gone, it's gone. In fact, I don't even think that, plants have, and we also have something called epigenetics, which is basically not your DNA, but the way that your DNA is used. So we have like chromatin remodeling, methylation patterns, these different ways that we express your DNA. And so plants do this a lot. And based on environmental factors, they can kind of like adjust to the environment or to what's happening. And part of what, we do in tissue culture is we reset that epigenetic profile. So if a plant had some sort of acquired knowledge almost or acquired reaction to a disease, it would almost be theoretically gone if meristem culture was done. So it should react like it had never seen it before. It should not be more resistant.
gotcha.
Oh wow, okay. So it's not okay. So you do lose the resistance.
Yeah,
that
I
you-
mean, there's if it had resistance genetically to start with, it would maintain that. In its actual DNA.
Oh
Like
man,
they
I keep
look all
lagging
the
out.
time for genes linked to like powdery mildew resistance. That was like a big topic that they did for a long time. And I don't think anything really came of any of those, genome-wide studies, which was kind of sad. It's really hoping for a gene linked to powdery mildew resistance that we could screen for, for outdoor crops or whatever.
Huh. We got another question from Tasmanian Devil. If you can remove a virus, can you alter, add, or remove traits also? I'm not sure what he means about traits.
So that would be engineering. So we would use genetic engineering to introduce traits. So that would only happen if we looked into that. So that would be where we introduced bacteria with DNA that we had put into it, or we used CRISPR or something like that. And it's not like we're affecting the plant's actual DNA. We're just removing the portion of the plant that is not touched by the viroid. So the viroid basically
Oop. Lost you for a sec.
hijacks the xylem and vascularized yet. I'm sorry, was that?
We lost you for a sec there. In the middle of the explanation. I think we lost her again.
You're going through a tunnel.
Yeah. So, that's a good. Tasmanian reminded me. Switch to live chat, everyone. While the internet figures itself out.
Meristem's the part of the plant.
All right. So it looks like you're back. So we're talking GMOs.
Yeah, so if we introduced DNA, that would be GMO, right? Like not like the cultivar, but a genetically modified organism.
Oh, okay. You think you
Stick
could
to the original
alter?
GM.
Yeah, I'm a GMO, but I like the strain, not the.
But the meristem, yes, same.
All right. Let's see. Oh, can you?
Yeah, not the
can
practice. Yeah. So
Damn. Well,
I don't.
I wonder if it's. I keep wondering if it's gonna catch up.
Yeah, I keep wanting to stop talking to see if it's going to catch up or not.
But I don't think it is. So, I guess we'll hit the questions after we figure out what's going on with the internet there.
Save your questions. Remember them. Write them
Yeah,
down. I'm gonna forget, so write it down.
I can mark them. So if you tag me, I can remember them.
That's awesome.
But yeah. So that's interesting.
That is
I kind of wanted to know that. Can you alter terp profile on a
I know
cellular
that.
level? Because man,
Super cool.
that would be some future stuff.
Yeah.
Oh, here comes Terpmeister himself. For goalie. Welcome.
Sorry, what Meister?
The
The Terp.
Terpmeister.
Oh, Terpmeister.
Yeah.
Oh, he
man,
said turd. Maybe he said turd. Maybe I misheard him.
Depends on your opinion, you know.
Uh,
Are you a saltiness only guy? You know, coming in swinging. Sorry, man, that's a bummer. I was listening and is finally able to jump on, and hopefully she's able to make it back.
Yeah, I hope she's able to make it
yeah,
back.
yeah. Hopefully just move a laptop or something if
I
possible.
know we were right in the middle of some good stuff, so
Yeah.
I kept thinking I was lagging. I was like, oh fuck, this is annoying. I want to hear this.
Haas Farms
Always
with
problem,
with
so I
the
was waiting.
Xfinity technical difficulty. Just sign out and come back. You'll be
Turn
fine.
it off. Turn it back
Yeah,
on.
turn it off, turn it on. But you know, sometimes that works. You never know. Like
I mean,
like
more
for
often
instance,
than
if my
not, you know.
internet's lagging like hers, I'll go unplug my router and my
Right.
modem. I'll wait
Yeah.
45 seconds, then I'll plug my modem in, let that slow mofo get to fully on, and then plug your router in. That
See
usually
all
does
those
the
lights
trick.
blinking.
But also, I notice on my phone, sometimes when I'm not getting good data, restart your phone. And it's almost like you reconnect and it gives you priority or something. I don't know.
I mean, it's just a small computer. We should be restarting them more often than we probably do. I actually have mine scheduled to restart every week, just in case I forget.
That's a good idea. I turn off my PC every night, so that makes sense.
Right?
I treat my phone like shit. I treat my PC like a queen. You
I know,
know,
same dude, same.
it's not good.
Hey, but if I could fit this whole setup in my pocket, I probably would treat it the same too, you know?
Yeah, that's true. Yeah. Welcome back. Welcome back.
Sorry. Hope it doesn't
You're
happen
good.
again.
It happens. It happens.
I was gonna say, Bake. There's these new routers and Wi-Fi systems and I don't know, software nowadays that you can actually like connect it if you have like an app or whatever. A lot of them have apps. You can connect it to your phone, just reset it like right from your phone, which is kind of cool without having it like it
Oh,
basically power
nice.
cycles it from your phone.
Yeah, I'm upgrading soon to like a Arrow or a mesh system,
Yeah,
so I
mesh
can try to
systems are
reach the
awesome.
pole barn.
Yep,
But
that's the only way I was able to do it.
yeah, so I mean, all I need is a little bit. I just want really honestly minimal. I need is operate them pulse monitors, you know, so I can make sure my grow is going. But
That and be able to like connect her Trollmaster is pretty sweet. But yeah,
yeah,
I mean,
like that's about the same level I would imagine of
it's
internet need.
yeah, it's those two things and then a security camera system, which I have.
Yeah,
Which
that's
is,
a good point.
that's what kind of drags on the data, you
Yeah.
know. But and yeah.
I measured it, and I had a buddy that got one, and he said his was a little bit further than mine, and he said so I should be good because he gets internet pretty decent. So I was like, all right, well, we'll see. So I gotta get that. That's on the list.
Yeah, another thing, right?
Yeah, exactly. I was just telling my mom, I got a list a mile long. Because they're like, we might visit, and I was like, well, sweet, I got a project list too. So,
Hey, you guys, let's bring a hammer and some gloves.
yeah, you guys ready to sand the deck?
Dude, that takes more than a day, just so you know.
Yeah, for real.
Oh well, if they're coming up, they've come up for a while, so
Look
I
at
gotta
the
put
weather,
them
too.
to work.
Dude, I had a whole list of
Yeah.
things to do today too, and I got a surprise one, a dishwasher that decided to stop draining on me.
Oof.
And it made me laugh because we were talking about that yesterday, about like how if you have the certain tools to fix the problem, you know, like you should be able to fix the problem if you have the knowledge and the time as well. And like, you know, screwdriver, some needle nose pliers, you know what I mean? It just doesn't take so much sometimes. Unfortunately, it's like I have zero knowledge, and I had to really go in depth with like the exploding diagrams of the parts and all this stuff to like figure
Yeah.
out where I'm putting things and all that. But yeah, the other thing I did was put up a trampoline, which was on my list of things to do today, and I was pretty stoked about that. Finally got a trampoline back we lost our last one in a massive windstorm, and took it out and threw it against our house, and then got some holes in it and whatnot, and ended up turning it into a added protection for the chicken coop and chicken run. which we were circulating some photos in our group chats in the last couple of days about that. That was pretty entertaining. But yeah, I mean, just reusing, repurposing, and all those things. But yeah, it's nice to have a trampoline back because man, it took me like a couple of months, but even my big ass was able to do a front flip. I haven't been able to do the back flip yet, but it's been a couple of years now, so I need to like get back in shape and maybe try it a few times before I start breaking myself.
Long time since I've done a backflip.
Yeah, maybe into a pool, but that's about it.
I feel like I would not be on a trampoline anymore.
No,
Yeah, I feel like that's a good way to go get a good lifelong injury. Like oh yeah, man. I thought I could do a trampoline. Now I've had a limp for 20 years.
dude, we're not that old. I've hurt myself worse disc
I
golfing.
still get on the trampoline, and I'm a boomer. It's not ever pretty when I get on the trampoline because moms shouldn't get on trampolines, but it's still fun.
I had a BMX bike and I took the wheels off and I would wrap towels around the spokes or around the forks and duct tape and towels around the forks, and it was a BMX bike, trampoline bike. Trampoline biking was a lot of fun.
Oh my gosh,
It was
dude,
very dangerous. Very dangerous. You're
What?
bouncing on the trampoline with a 40-pound bike, or yeah,
were you doing flips and stuff?
sort of, yeah.
At least attempting them. Dude, that makes me think of like the circuses or whatever, where they have like the cages and the motorcycles are just like doing these crazy, like, 3 or 4 of them in a row.
You pinch the.
You mean the cage
You
death?
take the seat off.
Probably.
The seat is terrible. Well, and I probably cut the seat on most of the time. But
I was gonna say, bro, that's real dangerous. Take the seat off.
All of a sudden, you're on a pogo stick.
I was gonna say,
No,
a pogo
no.
stick on a trampoline
when you
without.
take the seat off, you take the seat off, the whole post comes out with the seat. You don't just
Oh,
leave
okay.
it out there. You pull the whole post out with it.
Oh, if you, yeah.
And then
So-
how old were you?
Teenager.
What?
Well, you're a lot more rubbery when you're young. Correct me if I'm wrong, Rachel. That's straight science.
Yeah, I don't know if it extends into being a teenager, but yeah, when you're a kid, yeah.
Yeah,
Like a
see,
little
so,
Right.
yeah, so yeah.
You
Yeah,
just
well,
have
my-
house in your
They're doing
bones.
studies on seeing how long it extends. Some people believe teenagers, some don't. But let's
that
not get
is-
into semantics.
Your brain may not be fully developed when you're a teenager. Maybe why you're
Yeah, so
inclined
that's
to,
that. Yeah,
dive
there-
places.
yeah. Wrap a towel around the
Still
fork.
catching up mentally with the physical side of things, but it is funny to watch my 2-year-old, like, she gets tired and she just puts her face to the ground while she's sitting up, and her face is like next to her toes. You know what I mean? Like her foot's
Yeah.
right here. I'm like, I don't
Just
ever
holding her.
remember being able to do that.
Yeah.
You know?
Did you get her on the
Very-
trampoline and launch her yet?
No, well, I wasn't gonna launch her, but we
I
definitely
mean,
got
a
her on
mini
there.
launch, like you do it proportional to their age. But
Dude,
like,
she is
you know, boom.
such little like strength at that age that she's not like
How old is she?
she's just shy of 2 years old.
I thought, okay. Yeah,
Yeah,
that's pretty fragile, I guess.
Yeah, but also very spongy and rubbery,
but-
and that's the fun
but
part.
you
As
get
long
them
as she doesn't, like,
started
bite her
early.
tongue, she'll be all right. Like,
Yeah. Teach them a little bit. So I guess we can go back. We were
we have
let's
some
see, let's go
questions.
back. She was talking Mariston. You're talking Mariston, you're trying to get that out.
Yeah, I'm so
So let's
sorry.
do that. No, you're fine. Let's go back to that, and then I have to find the other question about terps. Because terps are always important around here.
Yeah.
They're-
Oh, the terp question was like, is there something that we could do to like affect the terp profile,
Yep.
right?
Yep.
So, I mean, short of genetically engineering, I don't think that you can, unless we're talking like changing the inputs to, modify the outputs. But they do look and find like genetic links to different terpenes. So, one of the interesting papers that was out recently was where they kind of debunked the indica sativa myth, but they instead plotted things based on their terpene profiles, and they kind of
Ooh.
segregated to that. And so I'm sure there's, obviously there's genes associated with the production of these different terpenes. And so you could screen plants to see. I mean, even this would actually be cool, even as a breeder, to be able to screen plants to see not just like how much THC or CBD it makes, but what the terpene profile is before you even, get to flower.
Yeah,
That
what
is it.
what kind of age, I guess, because you're taking like just a little plant sample, you could screen it at like day 3 or 4 or something. You know what I'm talking about, like a
Yeah.
seedling, right? And
Yeah.
that way, by the time it's, I don't know what, week 2 or 3, you know what kind of terps it has. Maybe it's
Yeah.
back from the lab or in this scenario, but that would be revolutionary.
Yeah, that's that's way more revolutionary and important than sex testing, than and to some extent even virus testing and things of that nature. I think terpenes are the most important part of this plant, and I will agree. I've said this probably a dozen times on this show, but a lot of the strains I grow are the strains I like to smoke. And the strains I like to smoke, after doing, plenty of lab testing, turns out to be high in not only CBGA or CBG, but also terpinolene. And both of those seem to be characteristics of our quote unquote sativa plants, like Durban poison, lime skunk, grapefruit Durban, clementine, like just this whole variety of strains I've grown and loved and really enjoyed and focused on for nearly a decade, I guess, at this point. What have you found though, Rachel, in your findings or the papers that you've read in terms of those terpene profiles and kind of how they correlate to those categories we put them in?
Yeah, I mean, they definitely correlate genetically. Those terpene profiles correlate genetically. The sad thing is there aren't a lot of cannabis genomes available. Like we talked about this last time, and I guess even like the chromosome organization of cannabis is poorly understood. I mean, there isn't the years of academic based research, to just kind of discover the basics to be able to like infer more or decipher more, so basically you have all these different companies starting at square zero, where they're trying to, do a massive genetic sequencing of multiple plants to be able to start drawing correlations, using biostatistics and things like that.
Hey, listen, we're catching up on like a 80-year ban on the science of this, so we're just happy to have you and people like you in our community, committed to something like this. But yeah, sorry to interrupt. Keep going.
Well, and it's even hard, it's an 80-year ban and it's still technically a ban. It's still federally illegal. A lot of institutions don't really want to touch it because it's a problematic,
Yeah, good luck getting a grant, right?
Yeah, and I mean, and even then, I mean, science is a institution where, the opinion of your peers is really important because they're the people that are approving your grants and they're the people
Oh,
that are
gosh.
approving your papers,
That's
reviewing
a
your
whole
papers.
we could do a whole podcast on that.
So, it's hard to step out of that like status quo for those people, I think. So yeah, so it is hard to catch up. And now it's great that we have all these private industries doing it, but then again, a lot of it's private, and it's not really widely available. So we're kind of at the mercy of whatever companies are doing that research at the time.
What do you think the next kind of groundbreaking testing that we'll do on cannabis is? Is it this terpene profile at a cellular level, essentially? Because that seems pretty groundbreaking to me,
Yeah. Yeah, I think the terpene profile would be really cool to see, like if we could just link the genes that make the terpene profiles to, and have an ID for those so that we could just amplify them, like we said. And in early pheno hunts, like you said, you could just eliminate anything that isn't going to produce the terpene profile you're looking for and only choose from the things that are just that terpene profile, and you're going to choose the best of what you already want. I was talking to somebody the other day and it was really cool. He was saying that somebody here around me was working on something where they basically get all the metabolic information, like metabolite information of a cell all at once. And like that would be really cool to see. You know what I mean? Because like we know of, you know, the terpenes, and we know about like the CBD, CBG, THC, you know, all these things. But it'd be interesting to see if there's even other like quieter players in that. You know what I mean?
So
If there's
explain
other,
that. What are metabolites? What are their function in this plant in particular? And you know, how would that help us as growers and consumers?
yeah, that'd be interesting. And the last time I was here, there was somebody who asked a question about like, what did I think about a certain company? And I wasn't familiar with that company. And I actually had looked at them once a while ago, and what they do,
Phylos,
what was it?
Phylos, the galaxy.
No, it wasn't Phylos. It
Oh,
was
or-
it was a company that basically they do cell culture, and they produce they do it with like pomegranates and grapes, and they're working on cannabis as well. And they try to get cells that produce the entire spectrum of polyphenols, you know, all the different things that a cell in that plant would produce, but in a cell system, or even more, like a higher level of those things, so that you could have like a healthier cell. And I mean, I guess to me, like all of those like cell culture constructs, to me, cell culture is expensive. Like whenever they're like, oh, fake meat's going to take over, you know, like tissue culture meat. And I'm just like, first of all, people have no idea how expensive like actual tissue culture is, like to raise cells in a dish. And second of all, to have animal cells live, you need something called like fetal bovine serum, which is from cows. So you're still like using animals to get that, you know? And so like with the cannabis stuff, I just think we're putting all of this extra input in to get the same metabolites out that we get when we grow the plant.
But explain. I'm sorry, I didn't phrase that properly. What like explain it like I'm 5, right? What is a metabolite, and what is
Oh,
it?
sorry.
Yeah, no, like pretend like I only took science 101, which I definitely did. Explain to me what a metabolite is, and like what is that doing for us?
Well, like all of the things that happen in your cell, you know, there's kind of like little machines everywhere working all the time. And so there's things that go into these machines, and there's things that come out. And these machines can be things like enzymes. You know, like acetyl-CoA in you is an enzyme that does something. You know, and plants have tons of these, you have tons of these, and they're constantly doing jobs. And you know, it's like with anything, you know, like when we looked at tobacco for the first time, you know, like one of the key things that we found was nicotine, right? And there's these big, you know, big molecules that come out of plants, and it's easy to just say, well, that's the entire part of the plant, and we're just going to extract that and package that, and that's all there is. But I think with cannabis, I think it's really nice because we see people kind of focusing on the whole aspect of the plant. And I think that, you know, when we look at all of these metabolites or end products of molecules, or things that the plant's making to protect itself, you know, or to have some sort of purpose in its life cycle, right? To make it attractive to pollinators, whatever it is. I think all of those things together kind of make the composition of the plant that's helpful for people. And sometimes I think taking one thing out is detrimental. So even if we do find out all of these metabolites or these end products, you know, even if you could put them all into a culture model where you just have cells that you're growing up to produce this, I still don't think it would have the same effect as like an actual plant. You know, just like a vape pen opposed to smoking the flower, right?
Exactly. I was just gonna say, and that is a very educated way of explaining why I don't like distillate. You know,
Yeah, same.
and honestly, why I asked the community if I should label my product as whole plant fresh frozen, WPFF, which is this new, you know, to me at least in the last year or so, a common term to describe the fact that you are essentially processing the whole plant. It's not just the larva, it's not just the trim, it's, you know, it's the whole plant, and that's
Oh,
98%
even the
of the time what I'm
families and
doing.
stuff?
Well, no, I mean anything with trichomes, I guess.
Okay.
Yeah, I am stripping off families because there's, you know,
Yeah, it's
no benefit to what I'm doing with those, but in terms of like I'm not shearing off sugar leaf, which you would do if you were trimming dry, right? And then like you have all this trim that then you might send off to distillate, right? That is not even getting sheared off, it's just getting put right in the freezer, put right in the tube and processed, and you know, But yeah, fan leaves, no. I try to avoid any exposure to like chlorophyll or fat lipids, anything like that. So I honestly, I try to cut my plants as minimal as possible with also breaking up like the nugs. You know what I mean? I want those nugs to be broken down node by node. But I don't want to expose just tons of like leaf structure and everything like that. That seems to be the best method for what I do.
So that's really interesting because when we do plant research in the lab, when we're trying to measure differences in RNA, which is kind of like the instant messenger that from the DNA to your protein, so it's the messenger telling your body or the plant what to do, right? What to make. And that change happens so quickly in plants that like when we take samples in the greenhouse, we take a sample and we instantly put it in liquid nitrogen. I mean, we have the liquid nitrogen right there because that change, once you damage the plant, is so fast. So it's interesting that like you found when I do this type of mechanical disruption versus this kind of mechanical disruption, I prefer the end product.
Well, and that's why I prefer a chemical disruption over a mechanical disruption, because it seems to be more controlled. You know what I mean? Which is again what you're doing with a liquid nitrogen. You know what I mean? You're using a chemical process to kind of freeze that state. Is it oxidation that you think maybe is contributing so much to the change of that, or is it because I can't imagine that the plant lives for much longer. I think about chickens, right? And they get their head cut off, and apparently they live for like a couple more minutes or whatever. Is the plant doing that? You know what I mean? If you cut off,
I mean, if you were to try to root that plant, would it root?
Just from the cut off leaves? No.
Well, not the cut off leaves, but like you cut the stem. And I mean, you can save that plant for a really long time.
Yeah, it might
still
take
get it
a
to
while,
root.
but yeah, you probably could.
So I mean, it's technically still alive.
Yeah.
You know, it's still doing its processes, right? It's still making RNA. It's still breaking things down. It's still having all those reactions and sending information through the whole plant.
So so that's another thing to think about. So when I do trim for dry, and I might be, let's say we're de-leafing, as part of the harvest, I will tend to cut the fan leaves off, like kind of in between the stem and the fan leaf. So along that, oh gosh, what's the word?
Petiole?
Yeah, I was gonna say petiole, but petiole. It's like terpinoline, or I don't know. I got corrected on so many of these different Latin words. You know what I mean? But yeah, when I shear off the majority of the leaf, and I go to dry it, it seems to be fine. It seems that that petiole kind of maintains some moisture, but not flavor, if you will. Similar to like a stem, it will hold moisture but not necessarily flavor. But when I have like a big old fan leaf and I attach all these fan leaves and I go and dry it like that, it almost seems like that flavor of a fan leaf, which is just not what I'm looking for, kind of gets sucked up into the bud. So my kind of way to balance that is cutting along that petiole, and that allows it to kind of dry a little bit slower, but also not kind of absorb that flavor. Does that make any scientific sense to you, or am I just like crazy?
I mean, like theoretically, we could say, phloem and xylem, just like when you make a cutting of a plant, they say like, oh, put it in water right away. Like if you're cutting flowers, you put it in water right away because you want to keep all those vessel elements open so that water can keep going up into the plant, or nutrients can keep, all that can keep kind of going up and back down. And so if you cut it too close to the plant, you aren't allowing it to make those plugs, those phloem plugs. So maybe you're just kind of like making a better seal because it has more highway space, if you will, to plug up and close.
Almost like you're clotting, right? If you're bleeding or something. Okay. Yeah. Okay. Interesting. Huh. But so
I don't know, but that would be my best guess if I had to say.
But if you left that fan leaf on there, aren't you allowing that highway to kind of stay open, and then maybe those
things from
starches or whatever they might be, those things that we're not trying to get in our bud, they might. Okay. So really we want to remove those upon harvest regardless.
I mean, I think it's a personal preference. Like, I'm never going to tell anybody how to, finish
Yeah.
their plants. But I mean, I think what you're saying, I definitely see some scientific basis to what you're saying. And I do think, like you said, you're not trying not to get like starches or different things back into the plant. So you want the leaf gone. But if you cut it too close, there's nothing to like to close, to seal,
Hmm. So what would be the how would that negatively impact your dry and cure if you're not able to close that? Would it just dry quicker essentially?
I
Or
mean,
just
theoretically, right?
yeah.
I mean, if you've noticed
Huh.
this, I would say like I would say if you've noticed this, it's a difference, right? If you've
Yeah,
been
for
able
sure.
to like observe this.
I mean, there's so many other variables, but yeah, I mean, yeah, that's why I do it that way when I am harvesting for a dried flower product because it seems to work for me. You know what I mean? And there's like you said, maybe some scientific basis, but I'm not a scientist. I'm just an experimentalist. You know what I mean? I'm just that's why I'm asking you these questions if it does make sense. Because if not, I don't want to do it. I want to find out a better way. But
I
what
mean,
you're
I don't think
saying,
so. I think all sciences is observing. And I think the fact that you're observing your plants and you can taste the difference between two different methods, I think that's all it is. Like you've decided this tastes better and that's what I'm going to do. And I don't think it matters if we understand why or not at this point. You know what I mean? And people can argue that all the time. People argue like little tiny methods all the time. And I think, sometimes it comes down to personal preference. But I mean, I think sometimes people just really pay attention to those subtleties. And I think you're probably just one of those people that has it dialed in and you're just paying really close attention to subtle details.
I got a Mac 1 here experiment. Number 1, 2, and 3 with different flushes to see which one's tastier. So I'm letting them cure up and then I'm going to give them a good smoke one like
How-
morning or something.
how many grams of each do you have, and can I get in on this?
So number 1, I had a bunch of grams. I guess I grabbed a big stalk. I don't know. This is all I have. And then
Oh,
number
that's
1,
plenty to share. Yeah,
yeah,
yeah, yeah.
yeah,
We can we
yeah,
can we can have a
for
little
sure. That's
conversation.
what I'm
Yeah,
saying.
come on, invite some friends over.
Well, I plan on sharing because that way I have the key on my phone. What number 1, 2, and 3 is. I'm not gonna tell
Nice.
anybody.
Yeah.
They're all Mac 1, and everything was the same other than the flush.
Gotcha.
So we'll see.
Hold on,
And
Rachel. Wouldn't we want to do a double blind study though? Isn't that only a
what's a
single
double
blind?
blind?
Oh, yeah, definitely want the double blind study.
That
Well, how
means
would I make a double blind?
that means you have now
No,
someone else
that's
has
what
to
Spartan
randomize.
suggested the other day. And I was like, well, no.
Yeah, we were
I
trying.
don't remember
The
what's
people
what
trying
though.
it.
Oh, so it's kind of like a blind and a half already study.
Yeah. I
Wait, if
I
you don't
put it
remember,
in
how is this scientific though?
well, I put it in the notes in my phone when I hung them. So that was like, week and a half ago. And I definitely don't know what's what. So I'm blind. I just double blind.
Well,
You blinded yourself.
I blinded myself.
What were the variations in your flushings for all three?
So normally I didn't do a water only because I never do water only. I normally do water and mammoth pea. So it's water and some microbes.
Mm-hmm.
So one of those is that. The other one is water and just canozyme. Supposed to help the plant get rid of all excess nitrogen, and that's the theory on that. Let it eat itself quicker, I guess. And then the other one's both water, canozyme, and mammoth pea.
Okay.
And so we'll see.
Okay, so real quick on that, you're getting very close. I know we talked about this before, but you're getting very close to the flush I used to do when I was in cocoa and running house and garden nutrients. It was a microbe, an enzyme, and a carb. So essentially, the only thing you're missing is like a carb or a hybrid type, thing.
Yeah.
Again, that was in cocoa, so I now my philosophy has changed that I'm in soil and things are a little bit different now. I think the enzymes are definitely going to help. I will say though that there seems to be I feel like I'm still testing this, and I don't have any kind of, again, scientific evidence for it, but it seems to me like running enzymes at the, in flush in flour is going to allow for the roots to kind of eat up anything else that's in the rhizosphere, right? But there's a potential for a lot of nitrogen to be stored still in those fan leaves. So that's why I think it's still important to maybe take some of those fan leaves upon the start of flush, whether you're using like an autumn flush or an enzyme flush or a carb flush or whatever, or water only, whatever it might be. It seems to me like it might be beneficial to take some of those fan leaves off at flush, because we're trying to get rid of the nitrogen anyway, right? For the most part.
Yeah.
So
I
it seems like
mean,
an assist, if nothing else.
I'm in there every night, so like if I see a
Well,
plant getting a little too bushy, I'll
that the
just
plant drinks
pluck it.
from those leaves.
That's true, but
You really don't need too many leaves for a thriving plant.
and again, those plants really slow down on the drinking at those last final stages, and yeah, you're right.
Yeah. I literally, that's another thing I've observed. 100%. I was just thinking about that at work.
And here's the other thing: if we're
you hit
flushing
like
with
day
enzymes,
59 or 60 or something, and the plant is not sucking water nutrients up like it was. It just shuts off overnight, and I'm like, all right.
That's
but
when you kind of know.
That's when I start flushing. Yeah, exactly.
Yeah.
I'm like, all right, you're gonna start getting a flush.
Yeah, that's usually
Yeah.
like, if you could time it right, in my opinion, that's like when like you should already kind of be in, like you should have started flush mode at that time, if you can time that right. And like, every strain's different and whatnot, but that seems to be what it's like. All right, I'm kind of done. Let's finish this out. I'm gonna take whatever's left in the roots, whatever's left in the fan leaves, and just throw it into the bud. And sometimes some strains really stack on the weight at that point. I've got a couple
in flush.
Yeah, man. And it's like, you kind of look at him, you're looking at him, and it's like, all right, like maybe this just wasn't it, this run, and then you start flushing and you come back like 2 days later and it's like almost doubled, and
dude, that's what got me on the Mammoth P too, was it made it even better. Like when I added, that to just water.
Again, because it
was
seems
like,
to be sucking the last of it up,
exactly.
right?
It's like and Mammoth P is designed to help, move phosphorus. So it's clear, it's doing something, because I'm watching the buds just swell every day or two. I'm just like, this thing, yeah.
So we talked about recharge and it, like kind of the focus on nitrogen on that. I wonder if I shouldn't, and I haven't been flushing with recharge or anything, but I actually have been kind of slowing down on my feedings with recharge and really trying to read the tips of the leaves, because I'm actually seeing a response now, almost like I did in cocoa, maybe like every other day, and how much I'm feeding them with recharge or just water only on some strains. So that being said, I wonder if I shouldn't switch to Mammoth P kind of in the last, maybe like post week 6, or maybe even in week 6 instead of using recharge.
I like Mammoth P, and the thing is that the microbe in there, as far as I know, is patented. So like, they're the only ones with that one that's designed to move phosphorus. So that's why I was like, and like it's like 0.5, 0.6 a gallon. Toss it in there.
Yeah.
Like I bought a liter for $120. It's gonna last me probably a year. So, and I'm pumping.
So you just made me think about this, and I don't remember what exactly the result of the conversation was, but we had Mammoth P on the Grow Tube back in the day. Like, they were a sponsor and all that. I got a couple like half gallons from them or something.
Oh wow, it's not I don't think it's supposed to go too bad.
No. Yeah. I mean, we definitely got through them, but one of the questions we had from the chat back then, and there was a big focus on microbes and whatnot, was, can you essentially breed these yourself? Like, can you put these?
I always have thought that, because everything I do, I try to get it to the do-it-yourself as much. Like when I had geckos, I bred isopods. After I found out it's like you can do it in a shoe container,
So I know they use a certain medium to breed theirs when they're, for their commercial scale production, but we were kind of like, hey, like if you just put an aerator in your reservoir with just Mammoth P, is that something that you could just kind of keep producing as long as you keep it? Yeah. So I don't really know where that conversation went, and I'm not I'm not fluent enough to give you guys any kind of real opinion on that, but maybe someone else can.
I mean,
Yeah.
if you wanted to culture it, if you knew what kind of organism it was, you could, easily kind of see what it feeds on and just like grow liquid cultures of it up,
But how does it stay good in a bottle? Like I've had this for months.
That's a good question.
So like that's what
They don't.
and
We're
could
dating.
you and Rachel, could you let's forget about the preservation fact. Could you just take some mammoth pee and culture it?
I mean, you should be able to. Theoretically,
Yeah.
if it's a live organism, you should be able to figure
Yeah.
out what organism it is and what it likes to grow on.
You just
and
can't
make
resell
plates and
it
liquid cultures.
because it's
Yeah,
patented.
hey, I'm using it for myself.
But you can
I'm
give it
using for
away.
myself.
You can use it for your commercial facility, probably.
Here's some micro smoothies.
There's this crazy old tech called
What you mean?
composting, guys, and you can make all the fucking microbes you want.
But I want the specific patented one. You know what I mean? It's
Yeah.
like really good for maybe CBG production, right?
Mhm.
If
Okay.
it's patented, they might have genetically
Yeah,
engineered
you wouldn't
it.
want
You
you wouldn't want
can't
the
patent things.
native microbes.
Yeah. So it might be, and if it's patent, if they've genetically engineered it, they might require a certain molecule to metabolize in order to live.
Yeah.
So you'd
Yeah.
have to know the conditions. You
It's
may
like the
not actually be
mushrooms
able
that
to
only
culture it.
grow under a certain type of tree at a certain type of year, at a certain type of client or whatever, or climate.
It's more like when we genetically engineer things, we engineer resistance genes in them or different ways to make sure that we're only getting what we want and not anything else. And so there's probably something in there like that, or there's something in there that helps them kind of keep maybe other people from culturing it up easily.
Is it are we focusing on pesticides more with that? Like not Miracle Grow. What is it? The one that we just spray on everything.
Glyphosate or
Yeah. Is that
Yeah.
kind of where the science went with that originally? And maybe now instead of trying to focus on like, hey, like let's remove the ability to get destroyed by this property and everything else around us can. But maybe now we're focusing on like, hey, how do we increase the profile or the potency or the certain characteristics, qualities of things that we want to like increase them, you know what I mean? Are we focusing on that at all?
Yeah, I think that actually was always the focus. I think the biotic resistance, whether it's like antibiotic or glyphosate resistance, those are tags that we add to things so that we can screen. So like to know that actually like our gene that we wanted in the organism was there, we give it a resistance gene and then we put that thing in that organism's world to kill it. So if it's glyphosate resistance, we use that glyphosate in actual petri dishes when we grow those different bacteria so that whatever doesn't have that resistance gene will die and whatever does have that resistance gene lives, and that's also attached to our gene of interest.
So you just made me think back to what you were mentioning about RNA and how they were more of a faster transport, and I hope this not to go political at all, but just in terms of RNA vaccines, now that's like a thing in my head, that I immediately wanted to ask if that was something that we could apply to cannabis, but it seems like we've already figured that out, because it seems like you could just tissue culture something and then propagate it from there without having to apply necessarily a vaccine en masse. But is that something that would or could be applicable to this plant, to maybe avoid something like hop latent viroid or something else that I'm sure will come about? And there's all these tomato and cannabis type hemp related viruses that will eventually crawl across our country, and in fact it seems like everything.
yeah it seems like just pathogens in all plants are up, in trees and plants and crops, all the things.
I agree.
So
But,
Rachel, would you be
it would
worried
be
about mushroom people then?
what's that?
Mushroom people like The Last of Us?
Oh man, yeah.
Like a virus like that? Maybe a fungal based?
Oh god.
Well, viruses and fungi are very different things, so it would be like a fungal based virus.
Yeah, it
But
would be.
but
It'd be
fungal
just a
pathogens can
yeah.
be vectors for viroids. So
oh, yeah. So they
Right,
had a
just
baby.
like bugs can be vectors
vectors, yeah.
for
Ooh.
powdery mildew, right? Like you could have an aphid or a ladybug or something get in your grow, and that brings powdery mildew or something crazy like that,
Yeah.
or a thrip, egg
Yeah,
or
thrips
something.
are piercing, so they're actually going to do some damage to your plant and put things in there. Yeah.
Mhm. Sons of bitches.
Put things in there. Oh. So we got hash time coming up. I'm gonna hit Rachel with some questions from earlier. So when we were talking about how you could track the terpenes in a plant for breeding purposes, you were anything Rose asked if you could track basically. Does that
That would
matter,
be so
female or
cool. Yeah,
male?
you could, right?
So
Theoretically,
like
just
yeah,
looking for the gene that makes the actual protein. Yeah,
okay,
absolutely.
because a big a big part of breeding is finding a stud. So like you could just
I didn't
and then
even
I
consider
guess
that when we were talking about it.
exactly.
That's
So then
huge.
you could choose a male and a female off rip off of like, well, they both have high this, so that's pretty cool. And then the second one, I guess this one.
Well, before you move
Uh,
past that, Rachel, make sure that you let us know as soon as you're able to do that.
yeah,
Right,
Yeah, let me go back to
seriously.
that
yeah,
paper and see if they mention those
You
gene
have so
loci.
many willing testers on this panel
yeah,
right now for you, Rachel.
for sure, for sure. The other questions: can you enhance terps? Like take something out of a different plant to put that terp into a strain.
And that's where you get into genetic engineering.
Mhm.
Okay, okay.
And I wanted to ask about that too, though. It's like, if we're already tissue culturing something and bringing it down to a maritime or a singular cellular structure, if you will, what does it take from a, let's go with layman's terms or as close as you can get, perspective of like how do we genetically modify a plant, and what does that mean? Because I know it's a very loaded and scary term, or at least it was like 10 years ago. I don't know if it still is, but a lot of us here on this panel try to focus like on the health and organics and bringing the medicinal value of this plant to the masses, really. And having something that's genetically modified does sound kind of scary, but at the same time, if we can maybe alter it in a way, or like we've been doing in many aspects, breeding it in ways to kind of lean towards something that we're looking for, whether it's maybe a certain trichome size or a certain flavor profile or something like that. What does it actually look like from a genetically modifying perspective? Like, what does that mean?
To genetically modify something means that we're inserting DNA that organism did not begin its existence with. So whether we're doing that using different bacteria, so for a lot of plants, we can use Agrobacterium tumefaciens, which will it's a bacteria that induces a tumor basically on the side of a plant. And when that happens, there's a genetic exchange that happens. So the bacteria will take plasmids that have DNA, genetic information on them, and ship those into the plant. And the plant kind of associates that DNA in like random places. And the problem with like traditional genetic engineering was we didn't have a good idea of where these genes were always ending up in the plant. And we also didn't really understand how many copies there were. And that's with animals too. Like when we talk about genetically engineering, like mice, but with CRISPR, when CRISPR came along, that allowed us to like specifically put in genes in places. And so that's a little bit of a different process. But at the end of the day, you still need a way to be able to segregate things that got that gene transfer from things that didn't. And so you still have to have some sort of a resistance gene. So if you're using bacteria and you're transforming bacteria, to get downstream, you're using an antibiotic resistance gene. If you're transforming, using that bacteria then to transform plants, then you're hooking it to like Roundup, a herbicide resistant gene at that point. So yeah, and I mean, I don't know, I feel like because we don't understand where these things go and it can disrupt other genes, and we're hooking on all these different resistance factors. I mean, I think the fact that we can just see what's happening is really cool. And I think selective breeding, like you're saying, is where it's at, because ultimately the plant is going to make the most healthy arrangement of chromosomes possible, not us.
Okay, real quick. as I
Oh,
drop my
hang on,
Deadpool-
Fergoli.
Okay.
So, if it's a genetically modified organism, if first, can something lose genes in time, like millions of years?
Yeah, absolutely. And plants even can kind of rearrange genetics within their own lifespan almost, because plants can do weird things with their DNA that animals don't do. So they can kind of like rearrange genes a little bit. They can have jumping genes, things that move around a little bit. So that is something that happens.
So if we found, this is just pulling numbers, making up numbers, but if we found a plant that was 1,000 years old and compare it to the same plant, but from modern day, and the modern day is missing a chromosome, this chromosome
Their
X
gene, yeah.
or gene, I mean, yeah. Can you would it be genetically modified to add it back?
Yes.
Yeah.
Oh, okay.
Unless you bred it with that plant. You know what I mean? If you had
Oh,
lineages that were
okay.
compatible, you could breed it back in.
Right. And that's what I
So
was
if
going
you,
to ask about the
oh,
organic method of genetic modification.
Yeah.
It's just essentially breeding, right? Like,
Yeah.
yeah. It's essentially what we've been doing with
okay,
dogs for
Yeah.
decades.
I
that
mean,
makes sense.
that way the plant is kind of deciding itself, like what's going to make it to the next generation. You know what I mean? Like when we rearrange chromosomes, it just seems to be unstable. Just like when we do reversals, a lot of times we end up with plants that are Hermes versus when we have a male plant that's pollinating a female plant.
I got to ask, how many chromosomes does cannabis have?
So that's super interesting because, to my understanding, and I could be wrong, but I did just recently really look into this, and it appears that like the chromosomes aren't well organized. So for every other organism, like you can look at a gene and you know exactly which chromosome it's on, you know exactly, where it is on that chromosome. You can look at all the pieces around it. You can get programs where you can just like zoom in any direction and see genetic code for that organism. And I guess for cannabis, the segregation of chromosomes isn't even like agreed upon. And so I guess the whole genetic structure is kind of poorly understood, which I didn't understand until I was kind of listening to another scientist talk about that, because the papers are just so like minor. There's just such a lack of information there to begin with, and I don't have like a scientific mentor that has been doing this for 30 years and is
Right.
telling me all of this.
Right.
I'm extrapolating from other areas of research to kind of put this together.
So is there nothing else out in nature that kind of mimics that? Do we not have, because we know we have like in humans, right? And even in other mammals, there's chromosomal differences, right? But to not be able to even like really map them, is fungi like that?
It's not that cannabis is like that. I think it's just that there isn't enough shared information, and maybe people have chromosomes listed differently. Like my private
Okay.
corporation has them listed differently from your private corporation.
Right.
Because,
So
when we do public research, it all kind of ends up in GenBank. It all ends up in this, public pool where everyone's using the same data to do the same research and the same projects,
goes back to 45 minutes ago about the peer-to-peer and all that. Like it's a challenge, especially with the
What's the law? The law gets in the way. man.
exactly the lack
Yeah.
of like regulation for 80 years on it, or ability to.
But it's
Yeah,
really
I
safe
mean-
to say that there's more combinations in cannabis than there are in humans. Like you can get more expressions in cannabis than you can with human beings as far as diversity goes. So that's just kind of
That's a lot.
mind-blowing. People say, well, why do we bother breeding anymore? Well, we haven't even seen the half of it, guys.
That's a
We haven't
lot
seen half
because
of it.
I just saw something that said, so there's 7 billion people on the planet, right? That's 7 billion different combinations. They did the math, and with our chromosome DNA or our DNA genes, there's at least 100 billion ways to make humans. So there's at least 100 billion people gonna be
Oh,
made
like
if
mapping the whole genome is what you're talking about.
if yeah if
like
you
the
rearrange them.
statistical.
Yeah. So if not more, so
yeah. I think
so
they.
that's crazy that cannabis has even more, combinations than,
I
which
think
means
thanks
we
to
still have a lot of GMO to hunt through.
oh my god. I think thanks to like 23andMe and things like that, we have done that very quickly. Unfortunately, I think it's cannabis might take a little bit longer to do that because I think we're creating new strains quicker than we are keeping on to the older ones. So it's almost like we're like the expanse of genetics is getting wider. The further we go, which makes sense, but it's almost like the tail end's getting deleted as we go too, and so we're not able to like make this catalog until we actually really like commit fully to like cataloging it. And like you're saying, Rachel, if we don't have like a common way of doing that, you might just end up with a lot of different graphs that you have to decipher down the road now.
We need a terpene galaxy.
So there is this girl, there's a scientist that I just found, and I can't remember her name, but she started an herbarium. She also does like analytical work where she's like doing pathogen screening and testing. But she started doing an herbarium. And if you don't know what an herbarium is, it's like a museum of plants. And so we go out into the nature or whatever, and we collect a plant in roots, leaves, flower, whatever parts are there, and you press it flat and you dry it out, and then you save it. And you can see the structure of the plant, the structure of the roots, the structure of the leaves, but you also have that genetic material. And so she's been doing a project where like it's a company and they're just building an herbarium of different cannabis specimens, which I thought was so cool because, how often do we lose genetics, or, we don't run something again and or it's gone, it's years back, and they always have this record of those plants. And I think that's so cool. Like I want my own cannabis herbarium at this point.
Can you give me a real quick Latin explanation of what the is in herbarium? Because there's arboretum, herbarium, like there's all the like aquarium, like all these that it's got to be like is it a
I think it's just
ecosphere or something? Like what does that what does that mean?
I don't know.
Oh,
I'm not a Latin aficionado or anything like that. But I mean, it has to be like some sort of enclosure, right? Like a sphere of enclosure.
Okay. I
don't know though.
Yeah. Okay. All right. Well, someone in chat maybe can look it up.
What if they dry them out? Can we still go back and snag a little DNA from it?
Yeah.
Oh, okay. And maybe revive it to a plant.
Or I mean, at least like be able to do a genetic profile, I mean, it's no longer with us. So unless you were going to do some sort of like way to use those genes inside a living plant, you know what I mean? Like,
oh, I see. You get the map, but you don't. Yeah. Oh.
But it
how
might
long
be
is
nice
that viable
to be able
for?
to link things, for linkage purposes. Like this gene showed up in this plant, and we've only kind of seen it there, and so we can look for that gene in other plants. We can
any and all data is gold when it comes to science. Let's be honest, because we can find ways to input it, make algorithms out of it, and just like extract some sort of result out of it.
Bro, they just found out gravity. They figured out gravity through math.
Per second
through math. No, they figured out that it operates on full scale, like as small as you can think on an atom, all the way to the universe size. It's like a circle, but they use math to prove it. I gotta find that video.
I hope that we're getting close, because it seems like the only way to get any sort of faster or close to or faster than the speed of light is through some sort of gravitational propulsion type system. You know what I mean? And that now we're talking like
they're already working on it. It's called the warp drive, bro.
No, I understand that we're really trying to understand it and work on it for a long time. It'd be nice for us to be able to use it. Is all I'm saying.
I think I was actually
just build these things, man.
Seeing an article that was talking that physicists were thinking that we were theoretically able to travel faster than the speed of light.
Yeah, but that's the time warping, gravitation. I'm not going to be able to even start explaining this, but Spartan looks like he wants to.
Well, I mean, yeah, the problem is you have to deal with time too. So, and then the human's body, being able to get through that successfully, we don't know what that's like.
Right. I think we
So there's going to be a lot of
theoretically can do it.
experiments, and I'm not going to be in it.
Yeah. I think we theoretically can do it, but can't maybe physically. Like we know that if we can kind of warp time and space at the same time.
Theoretically, it warps around you and you're fine, but
Right.
that's theoretically.
Yeah, you're
Right.
going a little bubble.
You're
and
like
then
To
that
you
physically
go,
move through that space and like come out as I'm looking right now on the other side. Who knows?
Well, I wouldn't mind. I'd probably let me see a test run and then I'll do it.
Yeah, I
Give it a
mean,
whirl.
You're going to believe some video.
Well, the thing too is where's the destination? Do
does
you
it
guys
really
have a cool
matter?
planet for me to go to, or is it just going to be desolate and I have to figure out how to eat and breathe and
That's
all
like
that?
Interstellar, right? Where they're just like world hopping.
Yeah, they
such
go
a
to the
good
one planet where a minute is
time.
like
Yeah,
70 years
dude,
on,
in those
he
waves.
just lost his kid.
Oh, I'm gonna watch that tonight.
I've been meaning to. That's a good one.
It's so good.
What, Rachel? What do you think about movies like that? Space science.
I haven't seen that movie.
Oh, it's pretty epic.
Dude, she is focused on a microscopic molecular
Don't
Yeah.
say
level.
that. I haven't seen that.
They don't have a lot of microscope movies.
Yeah, they really don't. What the?
I
Hey,
read a
you know
good
what? Hold
microscope
on.
book once.
What's Rosemary's Oasis? We'll get this. What was that? Actually, probably everyone on this panel will. What is that school bus?
The Magic School Bus?
Where we just there's a song though. Does anybody remember the song?
No.
I'm Bill on Capitol Hill.
No, she's talking about the Miss Frizzle.
I feel like Baked remembers it.
I know the show, but I don't remember the song. It's come to the Magic School. No.
Yeah,
I don't know. I don't know. I'll YouTube it.
I tried.
But I can't get the damn sound to go to the chat.
Oh yeah, that's weird.
But I'll sing it instead.
Just pull up those closed
Dude,
captions.
I showed you how to do that, man.
No, because I got to still switch everything over
Magic
to Google
Public.
Chrome.
It's a Magic Public.
Oh,
I think they remade that show.
See, there it is, the Mandela effect.
I wouldn't doubt it, Rachel. We're in an era of rehashing.
Well, that's true, but
And,
that's. I mean, is there anything else that kind of brings us down to that level of like looking inside of a colon? You know what I mean? Like stuff like that. Like,
oh, here it goes, Frigoli.
you
Next
got it?
thing that you know,
yeah. Nobody.
surfing on the Soundway. Yeah, I definitely would not remember that song. I was singing something a little different, but I remember all the characters. They're going through a body right now. That
I
was
really
a pretty
just wanted
cool
someone
episode.
to sing. I was hoping someone would commit, but I don't.
No.
I appreciate it, man.
Nobody wants to hear me
Yeah.
sing.
We'll take it, Tara. Anything I get.
sing
You
it,
remember
but
it,
I gotta know
Tara?
the
huh?
words. I gotta know the words.
Help them out. Help them out, Tara.
Oh no.
Haha. Yeah.
Here.
So I guess there's a really good documentary. It's older at this point in time. In fact, I feel like it was older even when I was younger. So it's
I'll
probably like
bring it up.
older now. But it was called The Private Life of Plants, and David Attenborough narrated it, and it was like a 6-part series, and it was really cool because like each part would talk about a different aspect of plants. Like there'd be a whole episode on just flowering, flowering plants, or
Oh, I just brought that up. Here we go.
yeah, I mean, and that's kind of cool because they're kind of starting to do CGI at that point in time, and so there's some really cool things to see in those documentaries. But I don't know that I've seen one recently that's like mind blowing in terms of like plant science.
He's also
Yeah.
I don't know if it's based on the book, but that book came out in 1995, The Private Life of Plants by David Attenborough. So I'm wondering if they made like a BBC documentary based on it or a miniseries based on it, or maybe they just came out at the same time or something.
This says 9 years ago. This was uploaded.
Oh no, yeah, miniseries came out in 1995 as well. BBC. I'm looking at it on Amazon.
Yeah, I want to say I tore into this like at least 15 years ago.
Oh yeah, there you go. See, guys, she sails the bay of pirates. Okay, no one else on this freaking panel knew last.
Let
night
me
what I was
tell you
talking
something,
about.
Fergola. The people that sail the bay, they just say the Pirate Bay because they're fucking pirates and they don't fucking have to use codes. That's what the
Alright.
fucking government guys do on the nice ships. I played tons of pirate games because I pirated them.
You know the game The Pirates on Nintendo, the original.
Yeah,
that
your
game is
favorite game.
so- yeah,
Dude,
it's
he
so-
got an Xbox that emulates old games, and you're probably still playing that.
I haven't played it in a while. I should play it again.
Yeah, that was a good one. I need to come check that out. You described it a bunch.
It's so dope.
Yeah.
I mean, graphics not great, but it's a fun game.
Yeah. So,
Yeah, there have been
anything
some
new?
really cool books out about plants recently, and just kind of different things that they do. It would be really cool to see maybe some new movies, CGI, like just like the mushroom movie that came out recently, to see that for
Oh,
plants.
Yeah, what was
fantastic
it?
fungi.
Yeah.
Fantastic
Fungi.
fungi, yeah.
Yeah. Yeah, the public's not a huge. The decline that went on like the History Channel and like where's the Discovery Channel anymore? You know, I don't think people watch too much of that stuff.
Well, it's all on like Netflix and stuff, like all those documentaries.
Yeah, well, that's
and-
yeah.
Yeah, I mean, there's so many David Attenborough ones on Netflix now. I think is where they're at. Maybe they're on. I don't know what's going on.
No,
with
nobody wants to
Rosemary
pay for
over
that
there.
bullshit anymore.
I
Put it
feel
on
you,
free
dude.
Streaming. That's
I
what
feel
it is.
you. Exactly. Sail the seas.
Yeah.
They just put it on YouTube for free.
Yeah, that too.
Oh yeah. The yeah, I forgot what I was gonna
Auto
say.
rotation. Rosemary's. He's got
I
his-
know,
Oh,
people are free
Rachel. What I was gonna say when I meant microscope movies, I was thinking more exciting like CDC has to, I once read a book where the CDC had to contain an alien virus from an asteroid. And they were in like level 5 underneath the ground. And it was very exciting. But like all I realized at the end like 90% of this movie was in a single room where they just were like, at a microscope and stuff. But it was very exciting. That's I'd like to see more microscope movies like that.
I think I probably have a hard time watching movies like that because they'll do stuff and I'll be like, oh, that's not even how I have that like I can't
Yeah,
suspend my
exactly.
disbelief.
True. Yeah, see, I know
Nerd.
nothing. I know nothing. So I'd just be like, oh,
Just like-
this must be accurate.
It's like Grown Weed has made all the weed movies like horse shit most
Yeah.
of them.
Yeah,
That's
yeah.
not how it works.
Here's the thing though. The Pineapple Express facility is still one of my favorites. If I could get
The
underground, like
under a barn in the middle of
yeah,
nowhere.
what in the what? That is awesome.
Yeah,
I
that
used
was
to be
pretty
like,
legit.
that's a dream.
Yeah, I'm the same way, guys. But I come from a little bit more of a I guess the production like videography side of things. So every time I see something like out of focus or like an artifact left behind or just that production value kind of stuff, that's what kills me. And so it really kills me when it comes to like paying for movies and you start seeing that stuff.
Like
And most
continuity.
that well, no, not necessarily so much like the plot or the organization or the anything like that. It's like it's literal like production value type stuff, and that's why like I have such a hard time. Like it's just like, oh my gosh, this is like
Well, I mean like homeboys holding a phone. They switch the camera to whoever's talking real quick.
and it's at his
In
he's not
5
holding a phone.
seconds, they switch back, and suddenly he's doing something else, and the phone's gone. It's like, well,
Yes,
what in the
yes.
hell happened?
Yeah.
Like,
Or the
who
positioning
who
is
edited
different.
this? Yeah,
Yeah,
like,
yeah, and
you
they
guys
they
didn't
shot
check that?
this scene 13 times and
Yeah.
it was 9 different
And
ways.
you guys won a Grammy or whatever they give
Yeah,
out.
dude,
What?
and like you forgot that he had glasses on once, you know what I mean? Like, oh my
Yeah,
god, it's stuff like
yeah.
that kills me.
Dude, they did a Steven Seagal is still making movies,
Yeah, of
and I
course.
saw a TikTok on one where they're like, "What the fuck, Steven?" And they had a stunt actor, and he had to be 100 pounds lighter than Steven. So like everything's silhouetted because it's dark. So you're like, you just know. Then the guy literally is in the light after the stunt for no reason. Just they decide to put the stunt guy in for the scene because it's kind of far away.
Yeah.
And it's so obvious. It's like,
dude. So speaking of, you brought up earlier Last of Us, right, on HBO. Have you watched the I don't know what they call it, like the recap, the production notes, whatever at the end?
what is that like?
Okay.
Like product? No, post
So
no.
at the end of every episode, if you're on HBO Go, what is it called now? HBO Plus something, I don't know, whatever.
HBO Go.
Yeah, the streaming one. If you keep it playing past the credits, it will show you like the producer's commentary or whatever. And they talk a lot about this kind of stuff and how like certain like the one episode where like the clickers become a thing and that the ground, well, hopefully I'm not spoiling it. Spoiler alert. The ground opens up
Yeah,
and like
spoiler alert.
a bunch of like whatever zombies or whatever come out of the ground real fast, right? They show
Yeah.
how that whole thing was shot and how they had to swap out the tank car that came in because they didn't want to blow that one up when they blew up the house. They had to create so many extra CGI bodies coming out and make them much faster. The bloater guy that they had was a guy in a suit originally, but he's just that big of a 200-pound suit or whatever. He couldn't move like this bloater was supposed to, where you're throwing people like you're no gravity at all, right? So they ultimately CGI'd that whole thing. Same with the little girl zombie that jumps in the car and it's like, hopping over the seats or whatever. They had an actual actress, like a paid actress, knows what she's doing, film the whole thing, right? Turns out it just doesn't give you that feeling on screen like if you CGI'd it. So there were certain things like that they went over in the I guess the producer's commentary or whatever, and they have some actors and they're talking about their experiences too, like what's his name, the main guy, when they're shooting in like Montana or something and it's like winter and they got these 2-story tall fans blowing on him and he's just literally freezing his ass off.
Yeah,
that's part of the scene. It kind of gives you that method acting part of it. So but yeah, that stuff is always so interesting to me. And so when it comes to even something like this, the production value of a live stream, right, it's just like like you got to bring it all together somehow,
For sure. Yeah. I'm real picky on that stuff. So it's hard to watch some movies. I just simply won't. I'll get a taste of it in the first couple minutes and I'll be like, oh, this is not good. But
it's kind of
like,
crazy because back in the day that stuff was like actually an art, like
well, yeah,
you would listen to the sounds of like everything that you did, like the sound of a teacup clinking the saucer, and like you would have to like make those effects happen in different ways, and you really had to like pay attention to what was happening to make it.
Well, that was also
filmed
before like laugh tracks.
I wish I could bring up a video with sound.
There's
a Facebook video I had on my Facebook that it was a chick that makes video game noises. And it's like, she literally has everything at her disposal. I don't know what company she worked for, but it was like, oh, you need snow, walking on snow. So it was like a really nice CGI video game close up of somebody walking and crunching some nice fluffy snow. She's stepping on charcoal, like kind of broken up charcoal, and it's perfect, and like all that kind of stuff. And like, it's like you said, I don't feel like the companies are working to outdo each other or like a pride thing anymore. Because I think I forget who I was telling this about, the video game industry. Look at the 90s. Look at Final Fantasies and stuff. Square Enix was showing off. Everything they did was first of all like it was like, holy shit, I've never seen a game look that good.
I remember the cutscene.
They were using orchestras.
Yeah.
For the music.
The music, yeah.
And now it's more just about get that game out or that movie out, and hopefully it sticks so that we can do a second, a third, and become a Fast and Furious. We're doing 10 or 11
microtransactions and pay to play.
Oh yeah, you want to see the DLC of the movie? Well,
oh god.
Yeah, but yeah, so I definitely agree there. I'd like to see some more just straight art, I guess you would say, of it.
I got
passion.
Yeah, that's a good point. Not just greed, just like everything else. But I got a BRB, but I want to leave you with this one thing that's going to ruin all video games and movies for all of you guys is any kind of metal industrial door closing. It is the same exact sound effect for every video game, every movie that.
I'm gonna Google that.
Yeah, you guys are gonna hear it once, and then it's gonna be the same every single time. You're gonna hear it like 5 times every movie you watch, 5 times every chapter of every game you play. Like
Now I have to find a game that has a damn door in it.
just
I was just
industrial
saying,
door
I
closing.
can't
like-
even recall.
Exactly, because you haven't
games
ever
or
thought about
movies
this
with that
before,
in it.
and now you will, dude. I think about the door, the coming in and out of the office on
This is a Reddit. Did you
Workaholics,
see the
dude.
Reddit? It's a huge Reddit thread.
Is it really?
Yeah, the squeaky door sound effect has to be an editor's troll. I swear to God, it's in every movie and TV show for the last 30 years.
Oh god. Okay. Yeah, I've never Googled this, but that's hilarious. I guess I'm not alone. I'll be right
Yeah,
back.
that's interesting. I'll have to. Now I'm gonna. Now every time I see a door in my games or something, I'm definitely gonna take note. I'm thinking of what games I have. Let me look. I gotta move this
Most
screen.
doors just have door, or most games.
Oh,
just
I
have
got Elden
doorways.
Ring. I got Elden Ring. That's got doors in it. I'm gonna check them. We'll see. I like facts like that though when they pan out though.
Yeah.
I wouldn't be surprised. I wouldn't be surprised. Oh yeah, you guys, the guy who made that door creak, bajillionaire, best
That was
door
my first
creaker in the
thought.
world.
Yeah.
He gets royalties forever. Like, we need a door sound. We got one. We got fucking one, and it's the one. You don't need any other door creak.
One
We
to
haven't had
rule
a
them
door creak
all.
made in 50 years. What are you talking about? This is perfectly acceptable. That's yeah. So let's see. I gotta pop a pack. I meant to go grab my seeds. I guess I can do that. I'll do that when Fergoli gets back.
What are you popping?
So that tray that I wasn't cleaning
Right.
is gonna be ready in like 30 days. So I should pop something like a single pack that I have, maybe a fem pack or I don't know,
You haven't
maybe
decided yet.
no, I just thought about it yesterday and then I forgot and now I remember it right
Pineapple
before
upside
the
down
show.
cake. That's my call.
Oh, those are fems. That'd be a good
Yeah.
one. Yeah, I gotta check it out. I'll grab my stuff and we'll see what I got.
And then I have for Growly make that concentrate. Oh man, dude,
Pineapple upside down cake, yeah.
that was so
Cause
good.
that
That's
pineapple, what did I have? Pineapple muffin? It didn't grow good, but it smelled good.
you had
Yeah.
didn't you have some kind of pineapple mousse or something?
I have pineapple mousse still. That's Skunk House. But I had
Oh,
pineapple
okay.
muffin autoflowers. Remember they were like
Oh,
black?
that's right.
They're
Yeah,
like so
yeah,
purple they were black, but they reeked.
yeah.
So I'm hoping that the pineapple upside down cake and that by itself is good. But I also
Good.
have that pineapple preserves from High Country. So
Oh, nice.
I don't know. We'll see. We'll
What's
see.
that? Pineapple upside down cake, man. That was some of the best concentrate I've smoked in a long time.
Yeah, that was good concentrate. Yeah, big decisions. So they're important because it decides your next 3 to 4 months.
Man, that blue dream's looking good in my garden, man. It's
Yeah.
dying. It's dying pretty quick. Like I think next time I grow it, I'll have to give it more food. But it's a beautiful plant.
It's interesting. A lot of those old school strains feed a lot more than any of the new shit I grow. Like the
Yeah,
sour
it's
D's,
crazy.
the New York sour D we had indoor. I grew that same thing, and the AJ sour D, and now the Blue Dream. So that's pretty cool. To like see a nice pattern in the older stuff being thirsty bitches, but yeah.
I still got a Project V going too right now. It's crazy. It smells like nothing. Like nothing at all. Like the Gorilla Grows Dream. It's tall, vigorous, and has no smell. I don't know if it's because everything else around it has a smell. It has nothing. Like I went up and pinched the bud and put my fingers directly in my nose, and smelled nothing.
That's yeah. That's why we need what Rachel was talking about, a terp profile, a profile calculator or something. And because I would laugh when that came up with 0 terps. They were just like, well, yeah, there's
I
your
do that.
answer. Yeah, it doesn't smell, but it also isn't gonna taste.
Yeah,
That would be weird.
But
That
if it
would be
still gets you
weird.
high,
Yeah, I
still
mean,
gets
if
you
you
high,
just
just
need
like
to get
smoking
that
air.
THC high, man, yeah.
It'd be really interesting. We should put it in the herbarium.
Yeah.
There you go, maybe I will.
Smash it in a book. Are they books?
I mean,
I think
they're
it's a
like
little more advanced than
technically
that
they're like
now.
plant presses, but I mean like it's 2 boards with some paper, like a thick matting paper almost, and you kind of like set the plant out and you squish it, and then like it's like belts almost that you like wrap around to like get the pressure to keep the pressure on them. But yeah, a book would do the same thing.
Well,
I mean, it's
where
smaller
do
though.
they
You can get
store
the
it?
whole
Where do they
plant.
store it after they smush it?
Usually gets mounted on a board and
Oh,
protected,
okay.
and then it gets like cataloged.
Okay.
And so, like most universities or botanical gardens will actually have an herbarium where you can go and see specimens of plants.
Huh. Like pull out a long file cabinet and there's just a bunch of
Yep.
giant slide, microscope slide things.
Well, I mean, they're big. They're,
No, the actual plant.
yeah.
It's
So you'd be
just
able
Well,
to
yeah.
like,
Well, I mean, yeah, but like in a large version of like, have you ever seen where you flip through all those
yeah,
slide,
yeah.
like Dexter, the serial killer guy? He had blood slide collection. And go. I'm imagining just a bigger version of that. That's kind of
I was
cool.
thinking butterflies, but yeah.
Oh, I
you're
bet they
talking
do
about
it with butterflies.
I
They
thought
do
you
it
were
with
talking about a
everything.
card
You've seen like
catalog.
Yeah.
jars of stuff, like kind of like
Yeah,
an image of jars with stuff in it. Same
Yeah.
thing.
things you can't smush, like a beetle, because it would just ruin it. You wouldn't want to smush a beetle and then mount it. It would just be
No, they
goo.
usually stick them and pin them.
A beetle would probably be fine, but it'd be like a mouse or something. Like certain things. I mean, I guess you could like
Oh,
stuff, but certain things you just maybe a fish. I don't know.
yeah.
Oh,
Oh,
Formaldehyde.
ugh.
A
Yeah,
human. I
you
mean,
wouldn't want to use it. So,
what was
for
that?
girly.
What was that?
Alright, I'm gonna go get my seeds.
Yeah, go get you those pineapple upside down cake.
We'll see. There might be better options.
There's not better, I'm telling you.
Everything in my collection is equally great.
We know it hashes well. We already know.
Well, we know what BHO's well. Well, I guess
Well,
that's
that's what
hash.
we're working
Yeah, I'm
with,
about to
man.
get that. Yeah.
yeah, okay. Oh, is there
I
a
knew
difference?
it's in the
name.
BHO extractor.
Let me ask for Gurley. For Gurley, do you think it would be a good idea to pop some pineapple upside down cake seeds?
That sounds very delicious and a great idea, Spartan. I think every one of us should do that on this page. I've grown Pineapple Express before. It was like definitely way early on when I got those seeds from like the Amsterdam store online kind of situation.
I've grown it too.
It tasted like pineapples. It had that kind of like hashy like I don't know how to explain that.
This
like
is way different. This is like the taste of a pineapple, like a juicy ass pineapple, and it's so good.
Okay, so does it have like that baked cake flavor or
No
just
cake.
like just juice?
I don't get the cake in the concentrate. We just had concentrate of it, and
Okay.
I didn't get the cake. To me, it just tasted like the fruit itself, like just the pineapple, like a juicy pineapple.
Okay, so they
But
got
yeah,
rid of
what
the rest
called
of it.
pineapple upside down cake. I don't. Maybe it's just a pheno of that strain. Maybe there's some more cakey phenos too.
Mm-hmm. That's what I'm thinking. They kind of like. I would be curious on the cross, but
Yeah, what is the cross on that baked? You're the guy on that. You're
I
my
wanted to
go-to
pull
guy.
that up because they also show their terpene profile on Humboldt Seed
Nice.
Company.
Oh, it is
Yeah,
HSO.
yeah,
Yeah, I grew HSO Pineapple
American.
Express, I think, and
Okay.
their Blue Dream, and I'm sure a couple of different ones. I always had good luck with HSO.
Upside down pineapple cake. Ooh, that's a mouthful.
I mean,
Not a good typer.
one bite at a time, dude. Yeah, chew 23 times or whatever they say, and then you can swallow it and then have another bite. There's plenty of cake to go around.
Effect. 40%. Oh well, Rachel just said they debunked indica and sativa, so this doesn't even matter. I'm not gonna mention that.
Well, the effects. So indica and sativa still work. I say we can save those words for adjectives when we're describing effects or when we're describing maybe plant
Right.
morphology.
Up or down, or basically.
That's
Right.
It
that's
says
right. You were there,
60%
Baked.
sativa.
Right when you're with the general public, like you either have
Yeah.
to say it's a terpene profile thing, like what flavor are you looking for or what effect are you looking for, and because they're going to tell you either they want sativa or indica or something else, you know, like I like citrus or I like your lime skunk or whatever, you know, but like I try not to put those words in their mouth. I really try not to. Sometimes it's hard because it's just we've been involved with it so long, but
And it's never in your best interest to argue with them.
No. I agree there.
Cookie Monster times Pineapple Trainwreck, which Pineapple Trainwreck sounds great.
I never was
Here's
a fan
the
of the train wrecks.
terpene profile from, I guess, one
Look
of
at
their
that
drugs.
osamine. Oh,
Yeah, a
man.
lot of osamine. I think that's why I
believe it's
bought it because
osamine.
I never
Look at
see that.
osamine.
But yeah, it's supposed to be pineapple and creamy vanilla frosting. But we got what we got was whatever pheno they had was straight pineapple. So that was nice.
Tangy creme brulee finish.
Yeah.
If
I don't
that's
know
a
what
fresh
that
frozen
means.
flavor I'm looking at, then yeah, let's do it. Scroll back down. I want to see that terpene profile again because
Yeah. I'll zoom in.
carrying. Okay, carrying. Interesting. There's
Some limonene.
no terpenoline. So I wonder if this is like short squatting photoperic 45 days.
Yeah. Hey,
What?
my buddy grew what was a blueberry muffin of theirs, and it was pretty close to 45. It was probably like 50. He pulled it.
Wow.
Yeah, so some of them I don't know, cause they breed for a while, but
Yeah.
you never know. That's the fun part about genetics.
Never know what you're gonna get.
Yeah. Let me see. I'll pull out some options.
45 days. That's a banger. If we get a 45 days, it's
Yeah, dude. 9 crops a year. Here we go.
Oh, I could do a sunken treasure seeds. What are these? Max Stomper or Stomp Perp? I don't want to do the Max Stomper. I've been doing a lot of Max stuff. I'm burnt out on Max stuff.
I was just gonna say, why don't we do something that everybody's not doing already?
That's true. That's true. Let me see. I'll keep flipping through.
You know, my
I
vote
don't think
probably.
I'm looking for Fems because I think I
I'm
just
thinking
want
of
to run
those
Fems.
stems we got from Humboldt that are called the pineapple upside down cake. That's the ones I'm thinking.
I heard you. Oh, well, I got
I
my
ain't heard nobody else
CSI.
running pineapple right now.
I have, that's true. Also, nobody's running Purple Urkel from CSI Humboldt.
You have straight purple Urkel.
S1.
Okay, that's okay. That's also a possibility.
I'm gonna put that in the possibility section. That's what I wanted to definitely. I like that one. All right, I'm gonna keep going then. So
It's
see,
not
now
gonna
your
be-
mind has been awoken.
Well,
to
it's
the
still not.
possibilities.
It's still not 45 days though. I'm
I
just
gotta
trying.
find the pineapple.
I'm
Here's
trying
the
to keep an
pineapple.
open mind. I'm trying to, man.
I taped it.
What's that one?
The pineapple upside down cake.
Okay,
Putting
yeah, that's
it in
beautiful, man.
Put in the contender pile. Man, we should have done a duck race.
You should do a vote. Can you do a vote in the YouTube chat?
Hold on. Before we get there,
No, I
duck
don't know
race
how to
would've
do
been
that on the
cool.
fly.
do you guys have any? And I know you guys are. Some of you guys are this age. It's gonna probably have to be the guys, unfortunately. But do you guys have Pinewood Derby cars from when you were kids?
No.
I won. I got to states with 1 year.
Do you still have
but
it, dude? You
I
guys
don't
still
know.
have it
My
somewhere.
parents might have it,
Call
but
your
I don't
parents.
think I do.
My parents my dad's trying to pawn all my old kid stuff off on me. He just showed me those while I was over there at Easter, and he's got like a whole box of them. I'm like, oh, we could do something with these.
Yeah.
What about a CO2 card? Does that count?
We could make
About
that happen too.
Melted strawberries. GMO times strawberry guava.
Oh, that sounds good.
Yeah, I mean, I could get on board with that. I like the
GMO, a GMO cross. I'm putting that in the contenders.
Yeah, 'cause that's totally new.
Surprise.
Nobody's doing. No one's growing any GMO or GMO crosses.
Oh, now somebody is.
Yeah, that's craziness. GMO and strawberries. That is outrageous.
A Florida man
It's
doesn't
gonna get-
understand sarcasm.
It's gonna. Oh, I do. I just you gotta pull through his because it's indefinite.
It really is. It really is.
That's true.
Such is your love for a GMO.
At this point, I will hear about pineapple upside down cake until these are popped. Even if I pop these first and grow these for 4 months, that's 4 months of. He'll come look at these growing and he'll be like, "That could have been some sweet pineapple."
Yeah, I
Or
would say I don't
something.
smell pineapples in this.
Yeah, something like that. I could see that. I could totally see that. Ooh, I got Clearwater Shock Tarts,
Ooh, that sounds
Pink
good.
Lemonade,
Ooh.
Pink Limonada, times Sour D Warheads, or SD Warheads.
Interesting.
Aren't you growing sour diesel right now?
I got it in the mom's area.
Mm.
I also have a POW, which is sour diesel times that warhead.
Nice. How many
Oh, another
active strains do you have or phenos do you have right now? Baked?
Why 12? That's all.
Yeah. 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 No,
He was thinking about it.
I got six Mac burgers. So there's 6 Mac burgers.
Yeah.
I got GMO, I got Mac 1.
Or even just different strains. I guess it's not even necessarily phenos, but you know, the
Different
more important
strains, I
question
probably
is
have like 10, 15, something like
okay,
that.
like how many would you like to get to is really the most important question because
I believe in freedom, so as many as I want, but
right. But in terms of scalability and things, like I as a solo
how many
caregiver,
moms do I want? I would say like 2 or 3, to be honest. maybe like, but I mean that's the thing. I know how to manage moms, so I'm not gonna. I can time it all right. So like that's only so that to get to the next harvest, then you chop that
Right.
big girl down
Right.
and reset her. But I mean like that's probably all I'll do because that's all I can fit.
Yeah.
too.
See, that's kind of where I'm at too. It's like I can only kind of keep as many strains as I have the room for. It's not
I
that
can reset them. I can clone them, get them to 1 gallons for a while, like literally, get them to 1 gallons for like 2 months,
I
and
just
then
do that with
reset
my
them again.
strains. Garlic
you
Punch
know,
is in 1 gallons right now. Cobra Milk is in 1 gallons right
and
now. Like that's the only
keep
veg I
doing
have.
that. And that's what I do at work. We do anyways. So that's what I'll probably end up doing. That's why like the tissue culture idea would be cool because it'd be like slowing that down a little bit. So it's not 2 months, maybe it's 3 months before I have to.
Rachel, do you
reset
have a
something.
storage solution where you can
Yeah.
continue to? And what is that like? It's like if I was paying like a like a basically a storage unit. Like if I was paying for one of those, is a similar kind of thing.
Yeah, we definitely put genetics back into storage, kind of to put them away. And in classes, we teach people how to store their own genetics safely to be able to pull them back out. Last thing you want to do is lose something.
Right. Kind of defeats the purpose, right? How long can you keep them in storage for before you want to really repropagate them, if that's the term?
I mean, like there's like by repropagate, do you mean like pull them out of tissue culture entirely?
Well, just saying. I mean, do you ever need
A couple
to?
months. No, I think like 3 to 5 months probably, and then I like to pull them out and kind of like just work with them a little bit and get them to the next level, like let them grow for a little bit and then put them back in.
And then put them back in. Okay,
Yeah.
okay. So yeah, it is maybe a 2 to 3 times a year process for every single phenotype, whatever that you might have.
Yeah, I mean, if you were gonna store them, yeah.
Okay. I just wanted to know indefinitely, like how long could you store them for? I guess, and is it like if we're talking liquid nitrogen and whatnot, like.
Yeah, I don't do the liquid nitrogen storage. Like I said, there's not really great protocols for that, and there seems to be like a difference sometimes in the way the the phenotype of the plant when it comes out, which is weird to me.
Okay.
But I've just seen these papers, and
Oh.
it just seems not.
Can I stop you? I'm sorry. What you just said though has made me think of something. I read a paper that hoplite and viroid in I don't even know if it was, I think it was cannabis, can potentially alter the monoterpene profile of that strain. Essentially, it seems like altering that DNA. Is that a possibility for something like hoplite and viroid, or maybe another viroid, to effectively alter the terpene profile of a strain, and or I guess temporarily or permanently?
So for an organism's DNA to be altered permanently, you would need it to be like a retrovirus, where you have the equipment to like take that DNA and put it back into the actual plant's genome. But for like hoplite and viroid, what happens is these small pieces of RNA interfere with molecular machinery in the plants. So it's very likely that it could stop something from being made. You know, you could lose terpenes based on whatever it's binding to. And I do think that they're kind of like getting better at locating like where that's actually, what that's actually affecting, which complexes that's affecting the actual hoplite and viroid.
Okay, I'm trying to search.
Well, it's not a DNA
Ooh.
change. Is what she's saying. It's something like when the virus is gone, the plant would go back to its original state.
Right. Unless
You
there was a
wouldn't lose it forever.
retrovirus. And
Okay.
a
Right,
retrovirus,
a retrovirus,
could
then it's a
permanently
permanent
alter.
change.
But
What is an example of a retrovirus? Or at least
I
in
think HIV is actually a retrovirus,
Okay,
which is
okay.
one of the reasons
so
why
that
it's so hard
alters
to
your
eradicate.
genes. Interesting. Okay. So I think maybe this is the paper I read, but the influence of hoplite and viroid HLVD infection on gene expression and secondary metabolite contents and hop glandular trichomes.
Oh, so it's in hop plants. And that's actually where a lot of the data for hoplite and viroid comes from, is hop plants.
Mm-hmm. So viroids are in the RNA. Is that true?
A viroid is a piece of RNA. It's not in the plant's RNA. It's like a piece of RNA. In fact, hoplite and viroid, some viroids have a capsule, like a protein capsule, but hoplite and viroid is just a naked piece of RNA. And it's a very small piece of RNA. It's different from the RNA that the plant actually makes, because that's messenger RNA. And then there's transfer RNA, and there's ribosomal RNA, but this is viral RNA.
Okay. So RNA is more just about the way it communicates, or the kind of type of
It's
NA it is. Okay.
So DNA is double-stranded, and RNA
That's
is
where we get the
single-stranded.
helix structure, right?
Yeah.
So single-stranded, it's just like a little worm spinning around, huh?
Yeah,
like a
it's
little
like
nematode
a
or something.
one-sided ladder, which normally is really
One-sided
unstable.
ladder, so it does have like the prongs coming off of it, though.
Yeah, because it's still the base pairs. That make,
okay.
Yeah.
So do those ever pair up? Is that possible?
Well, it won't like pair up with another thing, but
RNA.
it will match with different pieces of RNA in the plant, because it's like interfering with things that are happening there, right?
Okay.
But what happens with hoplite and viroid is it kind of like folds back on itself. And so it will kind of like have regions where parts of it kind of come back together and could like would be double-stranded, but it's not DNA, but they just kind of attach to itself. And what that does is it makes the structure really stable. So it's harder to break down.
Are there hobbyist or consumer level microscopes that we can use to view something like that, or is that just completely out of the
not a
range
viroid? It's too small for like, because we use like the visible spectrum, in a light microscope.
Mm-hmm.
And these things are so small, like you can't get enough light waves to hit it. So we have to use things like transmission electron microscopy or scanning electron microscopy.
So like microns.
Yeah, we're looking at electrons to see the shape of
that's crazy. That's so crazy.
Yeah, it's pretty crazy.
Those are probably $10,000 microscopes at least, right? Like
like hundreds of thousands.
Oh wow.
Hundreds.
Wow.
Like $500,000. Yeah. Like institutions, like an
Jeez.
entire institution is lucky to have one. And that's why
So
like
like a
big
university
institutions-
might have one kind of thing.
What's that?
Like a university, like a state university might have one, you know, if
Yeah.
that's like what their focus is.
Yeah. And like bigger universities might, like U of M had like a couple, I think, you know,
Okay,
but like
okay.
Eastern Michigan had 0.
Right. Yeah. Yeah, you're commuting for that class, I guess.
You're
Damn.
sending your samples over.
Oh, you don't even get to partake.
I don't know about that.
That's not if you're undergrad anyway.
Well, even, you know, even that equipment's so expensive to run, you know, like they're not going to let you run that equipment. You can sit there and like be like, "Hey, can you look over there?" Or, like, you know, and they'll but that's it. Like you prepare the samples and you give it, they work all the equipment. It's way too.
But hey, isn't there didn't MSU build like a miniature particle accelerator underground in Lansing or something crazy? Like
I didn't know, but that would be really cool.
I swear they're working on that. I don't know if that was if they ever finished it.
I'm on it.
There you go. There's got to be some news articles out there. I'm pretty sure I didn't break any
Confidentiality.
Yeah. I'm
don't
pretty
think
sure
we
this is like
got-
10 years old.
I don't think we
Yeah,
got
no,
the
that's
okay
cool.
on that one. I think that went to somewhere a southern state. I don't think it went to.
Oh,
us.
really? Oh, okay.
Yeah.
Maybe it was just the plans for it then.
A particle accelerator. They built a hospital.
Yeah, I mean, that's also helpful. Very helpful. I was gonna say keep it full, but that's like the jails. Like you don't want to keep a hospital full, really. Like
No, not at all.
it's good job security, but not really what you want to be secure on a job for, I guess. I don't know. It's weird.
What'd you find out, Baked? What's taking so long, man?
Says Lansing, Michigan State University, Center for Community Economic Development, Lansing Region Particle Accelerator Cluster. And it's like looks like a damn office park though. So I don't understand what that is. But then they go into particle accelerator. Oh, this just looks stupid. Never mind. See, this is the problem. You didn't give me prep time. I didn't find anything about Lansing.
It's called the MSU cyclotron, is what it's called.
Oh, I did see something on that.
Yeah, it's they operate 2 superconducting cyclotrons. They investigate the properties of rare isotopes and nuclear reactions. These types of reactions would take place in nature, in stars, and exploding stellar environments such as nova and supernova. So that's essentially what they're doing, is trying to create miniature supernovas underground in Lansing right now. So FYI.
Oh, they did that. They did that in
Cool.
China. They made like a little mini sun recently. That seems safe.
Yeah, just throw.
Seems like it'll keep it a little bit warmer.
if I could
I
just
don't
hold a black hole in my hand, you know, it'd be cool.
The
Right?
students are
Oh, they've
safe.
created a black hole. I can't remember who did that. Someone did just recently do that.
I know, that's what I'm saying. We're doing that kind of stuff too. Like,
Yeah.
it's freaking me out, guys.
Well, if
Yeah,
you want to
between
see
that,
the black
AI.
hole and the small sun, there will be Third Eye Craft Affair in Lansing this week, and you can come hang out from 11 a.m. to 5 p.m. at the Fledge, and come on and have a nice little sesh out in Lansing, everybody.
I'll be there too.
You think there's
find
gonna be
underground
bingo?
Particle accelerator while you're after it. I'm not sure if there's any. I'm not sure what activities are happening this week. You'll have to.
Mm.
The
honestly.
ground might swallow us all whole by 4:20 p.m., but we'll
That's right,
see.
by
I hope
4:20
you all
p.m.
see you
Huh.
there at the Fledge. Yeah, no, we'll be there too. I'm excited.
Opens up.
You got anything? You got all sorts of fresh stuff coming, right?
Yeah, we got a nice solid menu coming this week. Everything seems to be carrying up nicely. Rainbow
Is that
Drive,
what Becca's doing right now? She's
grape
working.
jelly, yeah, Death Star, Flow, Citrique,
nice.
strawberries and cream,
Nice.
Gorilla Breath, Hippie Crasher.
Damn, that's
That was
all
a
of
good
them.
one, dude.
That's all.
Yeah,
I loved the pre-roll.
Yeah.
It immediately started ruining, as the kids say these days.
yeah, really
And I was like, whoa. It was a good high.
I like the cake and punch too.
Yeah.
Cake and
Didn't you
punch
get
is
rid of
awesome.
that though?
Yeah.
You did? Oh, I didn't.
Yeah,
know.
dude. He got rid of that. That one was frosty as hell.
That was a good one.
Too nice.
I'll make sure I hang on to what I have then.
Yeah, I'm trying to remember the 4 in the last pre-roll pack I had. The party pack was, I know there was a GMO, there was a strawberries and cream, there's a hippie crasher at the top. I don't remember the other one though.
Maybe lemon dozy. Maybe
no.
maybe
I think it was something more like nighttime leaning. You know what I'm saying?
maybe gorilla breath.
It might have been Gorilla Breath, which Gorilla Breath is that knockout one. Which for me, Gorilla Breath negates the value of GMO. I'm sorry, Baked.
What?
You heard
What do
me.
you mean?
Wow.
Explain yourself.
Out of red/rosemary strains, if I had to cut one out of that 4-pack, it's going to be GMO because there's better options in there.
Oh,
I
did you
mean-
hear
I
that?
mean-
Rick? He said better options. You hear that,
GMOs.
Rick?
I mean,
Better
I can go to
options.
Webster's and show you what an opinion is, but everyone's got them, and that's fine. I would
We're all
not. I would not
subjective,
cut
man.
GMO out of anything.
But
It's the most unique, glorious strain out there. What are you talking about?
I don't know about that.
It's a
Yeah.
stepping stone for the future.
Yes,
Of-
something
of-
to step on to move on over and past.
Well,
Yeah.
that's why you got the crosses of GMO.
There's so many crosses.
You're telling me breath, and hasn't been
That's what I'm
used?
saying though. There's better options, you know. Like why have similar? Just kidding. I can't say that. I have so many citrus strains. I'm gonna shut up now.
Ooh, I didn't even think of that.
Red, don't get any ideas on cutting any.
I gotta get you some of this disco fries. It's kind of
Explain that
it
to
smell.
me.
It reminds me of your clementine
Really?
with like I don't know something else like a gas thing going on, maybe a burger funk. But that's what.
Okay.
Maybe a
Yeah,
GMO.
but it reeks.
That's all he's growing.
It's like clementine and GMO.
Well, maybe there's GMO. It's kind of like a GMO in the back, but like 90 to 80% is the clementine kind of smell. And to me, I wish I had some clementine to compare and be like, but to me, it reminds me exactly of that. And then, but like a little twist on the end. So we'll see how it finishes though. It's only day 55, 54.
What seeds did you pick, man?
Yeah, that was my next question.
I got two. So I put the GMO times strawberry back, not because of you, but because I
Oh.
realized I have a project pack of all GMO crosses. I don't know how I missed that one. So that went
Oh.
in there. So really it's down to, it's either purple Urkel S1 or pineapple upside down cake. And I don't know really how to decide. I was thinking about getting a coin. But
I know
oh,
what to
I
decide.
tell you what, I got a D20.
I like that, but
okay.
I want one more thing. I want to know one more thing because this might
Does,
be really crucial in the decision. How long does the purple
Does
Urkel
a
go?
GMO bag seed count?
Yeah,
There you go.
that's good shit.
What about
Yeah.
a GMO bag seed?
That's
got a couple of
feminized. So yeah.
Got a few.
So
What's
what
the
is?
flowering time on the purple Urkel?
I'd have to Google. Oh man, we have
It's
3
not on the
minutes.
pack?
Let's go. No, it's fucking. It's CSI. It just says purple Urkel.
Oh, go
Purple.
with the pineapple upside down cake. That's obviously better.
Yeah,
Pineapple upside down.
I
Okay.
do the pineapple upside down too.
45 days.
45 days.
I've
See, it's
had
Yeah.
obviously better, dude. They,
Well,
took it, they even
odds-
took the time. They cared
odds-
enough to put
Odds
it
it's
on the
purple.
pack.
Odds it's purple Urkel. Evens is pineapple upside down cake.
Okay,
God.
that's fair.
It's
That's
a
fair.
D20, by the way.
Is that your D20? Does it have your
It's
symbol?
the-
on
yeah.
it?
Base
No,
that count
no,
with it.
I can make mine.
See.
I'm rolling it on papers.
Right there. There you
Got
go.
the
What
rose
is it?
gold right
What is
here.
it?
It's a 3. I can't.
Oh no.
3. What did I say?
That means it's not the one I wanted, not the
It's
pineapple.
purple Urkel.
Yeah.
That's garbage. That's garbage.
Well, I have to go with the face.
Uh,
I'm gonna pop the purple Urkel to appease the gods.
The
You don't want
DG
to go
gods.
against them. Yeah, you don't want to go against them. But there's always next time, Spartan. This is only my indoor. We can do
Yeah.
a pineapple run with
What do you
all
got?
Ooh.
rainbow belts. Are you popping those soon?
Come
Oh,
on.
we only got 1 minute here.
Come on. Do it for me. Do it for me.
Yeah, pop those rainbow belts, dude.
Yes.
I want clones of them.
Please.
Rose gold is Pineapple Express Moonbow.
Oh,
There you go.
Well, why don't you guys do a pineapple off?
Heckle him, Spartan.
You could flip them upside down.
You want to do a pineapple off, Red?
Sure.
Yeah.
there we go.
Do that then, because
Yeah.
that makes it
Let's
interesting.
go.
I gotta pull those out then.
Free
Yeah,
processing
put
for
that
you
back out
guys.
But you're not gonna appease the gods, and you're not gonna get the best pineapple expression now, Baked. What are you gonna do?
I'll grow both.
There you go. Now he's got it.
There you go.
Had to get to minute 1 hour and 59 for that one.
Thanks for tuning in, chat.
With us today.
Tune in
Yeah,
for
Rachel,
pineapple
thank you for stopping by. It was a blast. Learned
Hey,
a bunch.
Rachel, can you shout out real quick where we can find you and your company and all that stuff?
Oh, yeah, I'm sorry. deeprootsttc.com. I'm on Instagram. Stop by, see me.
Yep.
Thanks for having me.
Leave your samples.
Yeah.
All right. Have a good one.
Okay, bye.